My name is Jeremy Testa, and I'm an associate professor at the Chesapeake Biological Laboratory, which is a member laboratory of the University of Maryland Center for Environmental Science, or UMCES. Thank you for the opportunity to present a subset of key findings from our team's research funded by the Ocean Acidification Program, which we have been performing over the past 5 years. I'd like to acknowledge my many colleagues here, including Jeffrey Cornwell, Michael Kemp, Ming Lee, and Lisa Wanger, also from UMCES, Wei Jun Kai from the University of Delaware, George Waldbusser from Oregon State University, and numerous students, postdoctoral scholars, and technicians. As you will see, an important theme in our research has been to understand processes that can mitigate ocean acidification, which I'll hereafter refer to as OA, in coastal waters, and to use numerical modeling tools to link an understanding of past and future acidification to risk and potential abatement in coastal shellfish aquaculture. Our funding responds to Theme 5 of the 2020 NOAA OA Research Plan, including the development of tools for managing coastal, open ocean, and Great Lakes ecosystems, and preparing human communities for potential OA-related changes. Our work, as you will see, addresses the goal to estimate anticipated changes to ecosystem services as a consequence of ocean acidification and evaluate alternative management options. We also will seek to test mitigation approaches under laboratory and field conditions and to develop adaptation strategies. We have received funding from 2 competitive OAP RFPs in 2015 and again in 2018, where we have used Chesapeake Bay as a study system to understand the interactions of fossil acidification, watershed nutrient inputs, and internal biogeochemical cycling and driving carbonate system patterns over both space and time. We've applied this fundamental knowledge derived from empirical observations and numerical models to identify actionable subfederal management options that can mitigate ocean acidification. As we've come to understand, there are myriad processes in the coastal zone that contribute to both enhancement and mitigation of acidification. At the heart of our original proposed research was a desire to understand at least a subset of what we hypothesized were the dominant controls on OA and estuarine ecosystems, including the competing impacts of eutrophication and fossil acidification associated with increasing atmospheric carbon dioxide. So to give an example, anthropogenic nutrients entering estuaries can enhance the magnitude of phytoplankton growth and decay, leading to pH increases in surface waters, but enhanced acidification associated with the respiration of that material in bottom waters. In eutrophic conditions, we have found that pH changes associated with the metabolism of phytoplankton are comparable to pH declines associated with local carbon dioxide uptake and exchange with acidifying ocean waters. We came to discover, however, that the simple competition between eutrophication-induced algal growth and fossil acidification was perhaps an overly simplistic view, given the diversity of exchanges and processes that occur at high rates in estuaries, some of which are illustrated in this diagram. So to give an example of one of the research programs we've executed, we did a comprehensive series of large-scale bay-wide cruises, Chesapeake Bay-wide, that combine detailed small-scale measurements around a macrophyte community with larger observations over the whole of the main stem Chesapeake Bay. And we came to understand that eutrophication, and perhaps its reversal, can lead to feedbacks that include self-buffering effects. So to tell this story in short, we first noticed an extremely large sink of alkalinity in the low salinity regions of Chesapeake Bay, which is indicated by alkalinity values far below what we would expect from the simple mixing of fresh and ocean water indicated by the orange line. We couldn't easily explain this alkalinity sink. So the sink was unexpected, and we identified that the region of alkalinity loss was dominated by a substantial, dense community of submerged aquatic macrophytes known as the Susquehanna Flats in upper Chesapeake Bay. An examination of high-frequency pH measurements within the middle of that macrophyte bed indicated that during our cruises, pH was often in excess of 9.5 and occasionally above 10, an extreme condition that would favor calcium carbonate precipitation, which would remove carbonate from the water, which is a key component of alkalinity and therefore a contributor to that alkalinity sink in the low salinity waters. We went to Susquehanna Flats and we did analysis of the materials extracted from the surface of the plant bed blades, and this indicated accumulations of calcium carbonate minerals that we noticed along those blades. We did subsequent modeling studies of fine particulate material that would look much like these calcium carbonate minerals and revealed that these minerals could be transported to the highly acidic downstream regions of the bay, indicated here where there's a TA source, indicated that those calcium carbonate materials would be dissolved. Thus, we've hypothesized that calcium carbonate minerals precipitated within these dense macrophyte beds can be exported downstream to acidic waters, buffering against otherwise more severe pH declines there. Given that macrophyte communities like these have been on the rise in Chesapeake Bay in recent decades in response to nutrient load reductions, our results suggest that nutrient management can reinforce naturally occurring buffering within estuaries to help mitigate against future acidification. We've also developed numerical models to address acidification mitigation strategies, where we have quantified the more conventional link between eutrophication and acidification. Here, the region of Chesapeake Bay bottom waters with extremely acidic conditions is indicated by red areas on the graph to the left. And the graph on the right indicates a large region of the Middle Bay, also in red and bounded by this green box, where the duration of acidified conditions is reduced by up to 20% with a substantial nutrient load reduction, indicating that these local nutrient reduction management can be used to mitigate OA as well. Our ongoing research, which we've started in 2018, is applying these numerical models to link historic reconstructions and future projections of acidification to potential habitat effects on the eastern oyster and the nascent aquaculture industry in the Chesapeake Bay region. While this research will continue for another year and a half, we have made progress in developing a linked modeling system to identify first the severity and duration of acidification and in response to elevated carbon dioxide, warming temperatures, and elevated river flow, linking those altered habitat conditions under future climates with the associated growth and mortality of the eastern oyster, and then linking this mortality and growth of the eastern oyster to the aquaculture community through economic modeling of the local industry and how growth of the oyster relates to that. So in summary, our research to date has identified multiple ways in which acidification may be mediated by regional and state level management actions. What's important is that these actions are firmly in place, involve regional partnerships, and include co-benefits. We have also shown that internal ecological changes can lead to self-buffering within estuaries, and these types of feedbacks are likely operating in other systems. They may also be leveraged to develop new and additional human-managed mitigation strategies. So looking ahead, I'd like to conclude that while discovery of internally generated buffering potential was not an intention of our original research, it emerged from fundamental measurements that NOAA supported, opening our minds to new processes that we would then build into our models. Secondly, although not highlighted here, our projections of climate change have identified both enhanced acidification and hypoxia in the Chesapeake Bay in the future, and that these two stressors may interact to negatively impact organisms and ecological communities. Understanding the dynamics and impact of these co-stressors in a changing world remains a challenge. Finally, although we have determined that existing state and other regional nutrient management is one tool to mitigate acidification and other stressors, the increasing pressure of climate change may require additional mitigation actions. And for example, engineered interventions that leverage new technology and ecosystem scale thinking should not be ignored as possibilities. Thank you for your attention.